The Th phenotype adaptive immune response is characterized by three key phenotypes: Th1, Th2, and Th17. Depending on the antigen presentation by the dendritic cell, that epitope that is presented to the T cell is recognized by the T cell receptor. With the appropriate co-stimulation, it will produce, for instance, cytokines like interleukin 12 and interferon gamma. These are the key two cytokines that will induce the differentiation into Th1 phenotype, which is characterized by a very particular set of cytokine production, which have the key target cell as the macrophages. This Th1 response that's actually conducted by the activated macrophages is basically triggered by intracellular pathogens, and it works in tissue repair and foreign bodies. In other words, the danger signal that is presented to the T cell by the dendritic cell is derived from either intracellular pathogens or because we do need tissue repair after surgery, after we cut ourselves, or foreign bodies, like having a splinter. So this is a typical response, and the mechanism by which the adaptive immune system is going to deal with that danger response in the Th1 uh, response would be phagocytosis. So they eat up the cell that has the intracellular pathogens. They kill the intracellular pathogens, and when they cannot deal with that in those ways, they can also induce granuloma formation, like in the tuberculosis disease, where if it cannot kill the intracellular pathogen, at least it keeps it on check by having the granuloma formed around the bacterium. With a different type of antigen presentation, we have interleukin-4 in the milieu that actually will differentiate that naive T cell into a Th2 cell. The characteristic cytokines of a Th2 cell would be different. You can see there's no overlap between Th1 and Th2 typical cytokines, and the target cell in this case is eosinophils. What makes the immune system become a Th2? Basically, it's parasites, allergens. This is where the drugs as allergens can actually also trigger a Th2 response. And this Th2 response will basically try to deal with that danger signal by producing an atopic response characterized by the presence of IgE, or antibody-mediated like the ADCC or the CDC complex of complement system to deal with the cells that are perceived as the danger signals. It also will induce mast cell degranulation. So this is a Th2 response. The immune system has specialized itself depending on the signal that's presented to the T cell into which kind of Th response they need to deal with a different antigen. And finally, we have a more complex milieu that will have a presence of certain cytokines like interleukin 1, 6, 21, and 23, but they cannot act alone. They have to be in a cocktail with TGF beta to then differentiate that T cell into a Th17. The characteristic cytokines of the Th17, again distinct from what we see in Th2 and Th1, will target the neutrophils. And what is the trigger for this Th17? Well, now we have extracellular pathogens where the neutrophils can actually produce antimicrobial peptides, or they can actually also help with barrier integrity, and they produce typically inflammation. Let's review the immune system in the gut. I highly recommend this paper, Nature Reviews. It's a very important paper that has a summary of the immune system in the gut. So let's take a look at what happens in the normal environment. The bacteria, which are the common cell bacteria present in the microbiota, they will actually produce a very healthy immune response characterized by certain cytokines like interleukin 22 and interleukin 10, but they are produced by the innate system cells. You don't have here adaptive immune activation. This innate immunity will help with maintaining the epithelial barrier and also maintaining a check on what we would call the autoimmune responses. The interleukin 10 is also very important in tolerance of certain signals. In addition, you have the presence of IgA, which are the typical secretory immunoglobulin that helps interact with the bacteria and the different products that the gut encounters. And again, this is basically all an innate system response, which is healthy and normal in the gut environment. So, in summary, the innate immunity maintains the barrier integrity and it is stimulated by the commensal bacteria. On the other hand, when we encounter any danger signal in the gut, that produces an activation and migration of the adaptive immune cells. Where there were no T helper cells before, now we see Th2, Th17, Th1 cells in the gut that are going to produce the typical cytokines now from the adaptive system, adding interleukin 17 and interferon gamma, and very importantly, activating the neutrophil. In addition, we also have the production of interleukin 4 from the Th2 and from interleukin 33 and TGF beta from the innate system will stimulate a Th9 phenotype that has been identified as playing an important role in inflammatory bowel disease. This Th9, interestingly, stimulates the differentiation and proliferation of Th17s. So the gut in pathology with the IBD disease especially will actually present an unusual phenotype that has all the important Th adaptive phenotypes Th1, Th2, Th9, and Th17 with a complex system of production of different cytokines. So adaptive immunity will be triggered in the gut by danger signals. These T cells, which normally are not there, will migrate, and with the appropriate homing of activated adaptive T cells, they go into the gut and they activate and produce the proliferation of innate cells that are driven by these CD4 positive cells. It's a very complex cytokine profile that is not predominantly either Th1, 2, or 17. It has all of them, and that's why inflammatory bowel disease is really challenging to treat, especially if you want to target a single cytokine.